Welcome, Rafa, and welcome anyone else who's out there watching. This is our second Google Hangout of our 2016 edition of Medical Neuroscience. And hopefully you got the communication that we sent out regarding how we want the next hour to go. So unless there are any questions about that, we'll just go ahead and get started. So Rafa, welcome. Thank you for joining us.
Thank you.
Nice to meet you.
Thank you. Actually, I was in the last hangout, but there were
Oh,
so
you
many
were? Okay.
students. So there were so many students that you probably don't remember all of us. So I'm Rafael from Brazil, and I'm a medical student here.
Oh, yes.
And
I remember.
I want to thank you for taking your time once again for doing this course for all of us. You are an amazing educator. And I actually have to thank you even more because I want to do my residency in the USA, so I'm getting used to the terms in English, medical terms, and neuroscience itself.
Very good.
I actually want to ask you if you could tell us about a new discovery that was once made by clinical studies that made it possible for new treatments for a specific neuro disease.
Do you have a particular disease in mind, or what are your...
Could you tell us about Alzheimer's? Because I think it's like a devastating,
Sure.
Is
Please feel free to interrupt me if you have a more particular line of questioning, but Alzheimer's disease is a neurodegenerative disease that we've known about for more than a century now, and it's been a very perplexing condition because it's not so clear really what are the principal causes. There seem to be perhaps different subtypes of Alzheimer's disease. There's early onset that often has a very strong genetic component, and that accounts for maybe about 8 to 10% of the cases that are known medically. So that means that the vast majority of people who have Alzheimer's disease are coming down with this condition without a very strong genetic component that would differentiate them from the early onset group. So that suggests that there's a multifactorial etiology that explains what's happening in these people's bodies and in their brains. So we continue to...
So I could have the gene but couldn't express it. Like, if is it possible that I have the gene but I didn't have Alzheimer in the future? Do you think so?
Yes, that's possible. So for example, the strongest genetic link that I'm aware of is an allele of a gene that encodes for a protein that is involved with cholesterol transportation through the body. It's called the ApoE4 gene. And if you have the allele number 4 and 2 copies of that allele, then you are more likely to develop Alzheimer's disease at an earlier age than if you have just 1 copy of that allele or some of the other variations of that ApoE4, or ApoE, I should say, gene family. So the genetic component at best seems to increase the probability that you might develop Alzheimer's disease. It's not an autosomal dominant genetic trait. So there is no certainty that you'll have it, but there'll be an increased probability if you have the right genetic conditions. So that suggests that the genes are not the whole story. That the genetic, your genotype, may vary the probability with which other factors, perhaps some are environmental, perhaps some relate to your lifestyle. For example, there seems to be some evidence that your aerobic fitness might offer some protection. Perhaps your basic metabolic state has some factor. But one of the very intriguing observations over the years that I think is quite fascinating is that we're beginning to think of Alzheimer's disease as not just a disease of the latter years of life, but really a lifelong disease. So there's some evidence that younger people, even in their 2nd and 3rd decade of life, are starting to evidence some motoric or cognitive change that we might be able to recognize as being predictive of the later development of Alzheimer's disease. So one of the interesting discoveries in recent years, to get directly to your question, has to do with the way the immune system interacts with the brain. So there's some evidence now to suggest that we should think more about inflammation as an important component to the development of Alzheimer's disease. So perhaps if you have a genetic predisposition, if you have a particular life history or lifestyle, and you have a particular trigger that might affect the way the immune response in the brain is activated, then those might be the necessary conditions for the development of Alzheimer's disease. So there's a lot of active research going on worldwide. I'm sure we will know much more as you enter the field of medicine, Rafa, and begin to contribute to that body of knowledge yourself.
Thank you very much for your consideration.
You're welcome. Thanks for joining us again. Good to see you.
Sarah should be next.
Okay, let's go to Sarah. Are you there, Sarah? It looks like your mic is muted, so if you could unmute your microphone. Oh, Sarah just left. So it looks like we have Ayush. Is that how you say your name? Ayush, are you there?
Yes. Hi, can you hear me?
Yes, I can. Go right ahead.
Hi, thanks. I really wanted to be a part of this video call.
Well, I'm glad you joined us. Where are you calling from?
I'm calling from Atlanta, Georgia.
Uh-huh. Excellent. So we just got a shout out from Nick, who's sitting on the other side of the desk from me. He has connections to Atlanta.
Mm-hmm. So I had a pretty specific question in mind.
Yes?
I was wondering, in many amnesia patients, usually they have no idea they can perform a motor task, but then when you put them in front of it, they can perform the motor task.
Exactly.
But I was wondering if it could happen the other way around. If you say, if they think they can't play piano, well, if they say they can play piano, but then they end up won't be able to play it if they have a cerebellar lesion, that would lead to loss of procedural memory.
Very good question. So let me back into it first by saying that if there were damage to the cerebellum, they still may be able to play the piano, but the coordination of the movements would seriously affect their competency or proficiency. So they would have a degraded capacity to play the piano. But it's not as if a cerebellar lesion could erase the memory for playing, let's say, a Chopin nocturne. So there would not be a loss of memory from that standpoint. There would be a degradation in motor performance. So that's the first thing that I would say. But to get to your real question, I think that's really fascinating way to sort of flip the usual way we think about this. So is it possible to lose the motor memory and yet still think cognitively that you've retained it? I'm not aware that that's possible. In some measure, because the motor memory or the implicit memory seems to be so widely distributed that I'm not aware that there is a brain injury or spinal cord injury that would specifically take out the memory without also degrading motor movement itself. So it would actually be very, very difficult to try to investigate or understand, because if a person's motor memory is stored in the motor system itself, which is what we believe, then it would be very difficult to tease apart their capacity to move and execute exactly the behavior that you're interested in recalling from motor memory. So that probably tells us something important about the difference between episodic memory, which is hippocampal dependent, and implicit memory of the motor variety, which is probably broadly distributed throughout the motor system.
Okay.
Now, of course, it's possible for individuals to have delirium, to have delusions, to have dementia, and believe certain things about themselves which are untrue. So someone may believe that they are a concert pianist when that might not ever be true. So that would be a somewhat trivial answer to your question, I think.
Okay, but that would lead from a lesion from a different part of the brain, right? Not really cerebellum or basal ganglia?
That's right. Or there could be a medical change in the brain related to medication, related to metabolism, related to glucose or oxygen utilization, something of that sort. That
So
may not actually.
more of a like a biochemistry kind of problem, more than a
Right.
lesion per se.
That's right, as opposed to a focal disturbance. Now someone potentially could have a focal lesion somewhere in the prefrontal cortex, the front part of the frontal lobe,
Yeah.
and affect their beliefs about themselves, but that would be fairly rare. Usually when people have these breaks with reality due to delirium, psychosis, schizophrenia, dementia, it's a much more diffuse problem, not really a focal condition.
Okay. Thank you.
All right, you're welcome. Thanks for joining us. Okay, let's go to Juan Carlos. Are you there? Juan Carlos, your mic is muted. Could you unmute yourself if you're still with us? Juan, it seems like you're here in the chat, but we do not hear you. Okay, I think Juan Carlos just left. So there are plenty of spaces available in the Hangout at the moment. So if you're in the first group that we've queued up for this hour, feel free to go ahead and join in. I suspect some of you are out there just watching, and that's fine as well. But I'd love to have you join in and share your thoughts or your question or your comment. While we wait for somebody to go ahead and do that, I hope you're enjoying medical neuroscience. I hope it's going well for you. And, okay, so it looks like Nick's telling me we have some questions. So Nick, go ahead and let me know what you got.
All right. Does damage to C4, C7 disc between, which is pressing on nerves, revealed over time after an accident, and can steroid injections cause a patient to become imbalanced even after several sessions? What are activities or changes in the brain which causes this imbalance reaction when it comes to steroid injections?
That's a tough question to address, very, very complex. I can imagine that if the steroid is somehow elevated throughout the central nervous system, that's one set of effects. And it depends upon what kind of steroid we're talking about, of course. Otherwise, one might think about there being some local impacts at the site of injection in the lower cervical cord region. So there could be mechanical effects of the injection procedure, but setting that aside and thinking about the interactions of a steroid hormone with brain cells, one can think of there being possible ways that that steroid might affect both the neurons and the non-neurons. So usually steroids are used to try to control inflammatory processes. And when it comes to neural tissue, we know that affects the way some of the glial cells interact with the tissues that they surround and support. So one might be concerned with whether there may be some activation or modulation of glial cell function that might have an impact on neural signaling and perhaps even neural structure itself. So those would just be some thoughts. I'm afraid I don't really know too much about that particular topic, so I should probably not speculate too much beyond my knowledge base. Okay, let's go to Hadi. Are you there? Hadi Zadeh? Okay, let's go back to Sarah. You were with us a few minutes ago, but your microphone was muted, as it is now. If you're there, Sarah, and you'd like to join the conversation, please unmute the microphone. You can do that at the top of your window. Okay, it doesn't look like Sarah is doing that, so how about if we go back to Rafa, who's jumped in again? Are you there, Rafa?
Yes, I am. I'm sorry, but you asked for us to return if you were alone, so I'm here again.
Very good. Glad to have you.
So I actually have one more curiosity that I actually want to ask you. If you could tell us about the neuroscience behind dreams, could you tell us about that?
Behind dreams. Yeah, that's a fascinating subject. Um, we still don't really have a satisfying answer, I'm afraid. One aspect of dreams that seems to be um, really amazing to me is that while we're asleep, in REM sleep is typically when we dream, and yet if one looks at the cortex is remarkably active. This is why REM sleep has been considered to be paradoxical sleep. So clearly there is a lot of activity going on in the cerebral cortex, and that's very likely where the dream is being elaborated. Now, the trigger for the dream is still not entirely clear. There's an electrophysiological phenomenon called geniculo-occipital waves that potentially could be the trigger for the actual brain state that produces the dream. And that electrophysiological phenomenon is coming from the brainstem. It impacts the lateral geniculate nucleus in the caudal part of the thalamus, which then projects to the visual cortex. And this very likely explains why so many dreams have a very profound visual component to them. So it may be that the visual system is the first of the systems to be engaged, but once that happens, then processing can spread throughout the associational cortex, and one can engage other systems, other sensory systems, other cognitive systems. Quite often, dreams have a very profound emotional component to them, and that very likely comes from activating structures like the amygdala in the medial temporal lobe, as well as the orbital and medial parts of the prefrontal cortex. So the content of the dream is another fascinating component to your question, and that probably reflects the kind of activity that is being sustained within these cortical networks. So we often dream about fragments of what has happened in our lives in the recent past or perhaps even the long-term past, and that suggests that there's some kind of memory component to the dream. Now, a very interesting aspect of sleep research in recent years has been to recognize that we actually learn while we sleep. So there's a kind of replay that's going on while we sleep that might help consolidate the plasticity that underlies learning and memory. So I think it's a fascinating question to know whether dreams have any role in that at all. So people have wondered for centuries, literally, whether there is an adaptive role for dreams, and we still don't have an answer to that, but it could be that something about the dream is facilitating the consolidation of learning that's happening in our brains while we sleep. So those are just some thoughts. I hope that's helpful to you.
Yes, it is. Thank you again.
Okay, it looks like Hadi is with us now. Can we go?
Yeah. Can you hear me now?
Yes, I can.
Okay, great. I just had a thank you note here. I'm in my office from San Diego. I'm in Google. So I just wanted to admire and appreciate that you have this course. I'm following it, and it's very, very good. I'm really excited to be part of it. And as just a side question, you may or may not answer this. I just was wondering that if you have a plan to cover addiction or any compulsive related behavior in the brain that might tie into addiction in your course.
Yes, first of all, thank you for joining me in medical neuroscience, and thank you for your kind words. That's very encouraging to Nick and myself and others involved with the course. Addiction is covered very briefly in the very last set of tutorials at the end of Unit 6 of the course. So there could be much, much more that I could or perhaps even should say in medical neuroscience about addiction, but that's where you'll find it. There are some comments about addiction as we go through Unit 4, particularly when we talk about the basal ganglia and the ventral stream of processing in the basal ganglia where motivated behavior seems to arise. So that seems to be ground zero, if you will, for drugs and experiences that have addictive properties in the brain. So elements of the circuitry that is important for addiction comes up in Unit 4, and then we return to it at the end of Unit 6 after we say a little bit more about emotional processing in the brain. So medical neuroscience is going to be revised later this year, and addiction is one of those topics that I will expand upon in the next edition of Medical Neuroscience. So hopefully you might consider joining me again in the future, and we could talk more about addiction at that point.
I don't mind at all. Thank you very much.
You're welcome. Thank you. All right, it looks like Juan Carlos is here. Juan Carlos, if you're with us, please unmute yourself and go ahead and ask your question. Okay, maybe I'll come back to him, but I see that Bilal is here. I hope I'm pronouncing your name correctly, Bilal.
Hello, can you hear me, sir?
Yes, I can. Nice to have you. Go ahead with your question.
Sir, first, I would like to say thank you for giving us your knowledge, and also thanks to your entire team.
Okay, thank you.
Sir, I am 22 years old, and I'm a computer science student, and I live in Pakistan. So I took this course because I want to learn how brain works and how do you think fast and take decisions. And you know, I want to go inside my brain. I want to learn everything. And my question is, I have one brother, and my brother is 3 years older than me. And if I am thinking something, then before I speak, my brother speaks same what I thought. I want to know, is my brain connected with his brain or what?
That's a great question. In a manner of speaking, I would say yes. In a manner of neurophysiology, I would say no. Your brains are not connected anatomically or physiologically. But the way in which I would say yes is that as we become familiar with individuals, particularly family members, we are receiving information from their entire person. That includes what they say, but it also includes how they look at us, the movements of their eyes, the movements of the face, their body, and we are reading these things instantaneously and building up a mental model of what that information is conveying. So it's very possible that your brother and you have such experience with one another that you are reading the non-verbal aspects of language that you're communicating through your gesture, through your face, through your eye movements, and you are already building a mental construct in your mind of what is going on that might lead to the next verbalized thought. So in a sense, you may be reading one another's mind, but what you're doing, I would suggest, is that you're reading their gestures. And there are gestures of hand, of face, of eyes, of entire body. And so one of the things that the brain is really good at is making predictive models. And as a computer scientist, I think you can understand how important that might be in communication. So if you're building predictive models, you may have a sense of what is about to be verbalized, especially by someone that you know so well, such as an older brother. So I guess that would be my speculation to your question.
Thank you. Thank you.
Okay, very good. Thank you, Bilal.
Dan, what's next?
Okay, let's go to Mario. Are you there, Mario?
Yeah, can you hear me?
I can, yes. I don't see you, but I can hear you.
Here, because I'm currently in the library. I wanted to ask, this is not like a very in-depth like neuromed like question, but it's more like a general one.
Yes.
Regarding medical school in general. I'm currently a sophomore. I'm studying at university here in the States, but I'm an international student, and I've been like a little bit discouraged by the fact on how the quotes are with international students to get into medical school. But I want to give it a try, and I'm interested in like neuromed. So what are some tips that you'd like suggest me to like stand out among like some of the applicants so that like my chances are, let's say, like a little bit higher than the others? I'm currently not a freshman, I'm a sophomore. One thing that I was thinking was applying for like a master's, then doing like med school.
Yes.
So what would you say about that?
Well, that's possible. One of the challenges is not so much medical school, it's whether you intend to seek residency training in the United States or whether you might go to your country of origin. So most of the residency programs in the United States have restrictions on who can fulfill those residency slots. They're mostly funded by the United States federal government that would support um, United States citizens and permanent residents. So many medical schools have a bias in favor of United States citizens and permanent residents. If you're not in that category, it's not impossible, as I suspect you've already figured out, but there is a bit of a higher standard. So one way to distinguish yourself is, of course, to have excellent academic preparation and have that manifest in an excellent medical college admission test, or MCAT score. You should also distinguish yourself by research, getting involved in biomedical or health science related research, and then distinguish yourself through the depth of your exposure to the healthcare setting and the health professions. And that might involve shadowing physicians and nurses and therapists and other kinds of healthcare providers, but it might also involve volunteering in various ways to demonstrate that you understand that a life in medicine is a life of service, and that you can do that, and you can do that well in a variety of settings.
Another thing is, I've been having this discussion with some people as to how valued a bachelor's degree is when it's a BA or a BS. Would you say that like a BS is like more valued than a BA, or should I aim for something like a BA, have higher grades, which will like be sort of like a better CV rather than go for a BS and have slightly lower grades because of the difficulty?
I can tell you the Duke University answer. The Duke University answer is that we do not care whether you have a Bachelor of Science degree or a Bachelor of Arts degree. What we care about is what you have done. So if you find a Bachelor of Arts degree as what you find the most fulfilling and compelling way to experience university, then you should do that, but also attend to those other issues that I described, getting involved in research, taking challenging science courses that will prepare you for medicine. But you can do all that as a Bachelor of Arts student, majoring in whatever discipline interests you.
Yeah, okay, I understand that. That's like a perfect answer. Thank you.
Okay, all right. Thank you for joining us, Mario.
Thank you.
Okay, let's go next to Dan. Are you there, Dan? I see you, but you are muted.
Can you hear me?
Yes, I can hear you now. Thank you, Dan. Go ahead.
Thank you very much, Doc. My name is Danny. I'm from Kenya. I'm probably the only student from Kenya.
Welcome.
I guess I can outrun you all in the Saturday morning runs.
Okay. What city in Kenya are you in?
I'm in Nairobi.
Nairobi. I was in Nairobi briefly a few years ago.
Okay. My background is in medical biochemistry and clinical psychology. It's quite wide, which
Yes.
explains why I'm so interested in neuroscience. But my question is a little bit controversial because it's all about neuroenhancement. The role of neuroenhancements on neuroplasticity. And when I say neuroenhancement, I mean reference to tropics, piracetam, modafinil, and the rest. So
Yes.
if you could maybe shed a little light on their role in neuroplasticity, I would appreciate a lot.
Sure. Well, I don't have too much to say, Danny, specifically about any one particular agent, but I can tell you that the mechanisms of neuroplasticity can be modulated by a number of different biochemical and cell signaling pathways. And it is very likely that we will find a variety of pharmaceutical agents that will enhance the plastic response to a variety of signals. Some of these are genetically based, so you may know that one of the key components to mediating structural plasticity in the brain is the interaction of brain-derived neurotrophic factor with its receptors. And some individuals have genetic variants in their receptors for BDNF that enhance plasticity or perhaps make plasticity a little less robust. So that's an example of a genetic variation that can have an impact on learning and plasticity and perhaps even skill acquisition. So if we can find agents that might similarly modify the brain-derived neurotrophic factor interaction with its receptor, we may have an opportunity, therefore, to enhance plasticity in certain contexts. Some are trying to do this by shifting the balance of excitation and inhibition in the brain. And if that balance can be shifted ever so slightly in favor of excitation, then that might make long-term potentiation more likely to occur. Another possibility is to use other kinds of agents, including things like electrical stimulation or transcranial magnetic stimulation as a way to enhance plasticity. So I think some combination of pharmaceutical intervention with some structure in the sensory and motor experiences of the mind and the body, together potentially with one of these physical agents like nerve stimulation or transcranial magnetic stimulation, might actually enhance plasticity in the brain. And this might be quite valuable for individuals recovering from brain injury and disease, but it also may be useful for individuals with intact nervous systems simply to enhance their performance.
That's right. Okay, thank you very much. I'm actually doing a literature review on the same. I'll probably be having a paper out by the end of the year.
That's wonderful to hear. Thank you so much, Danny, for joining me. And it's always wonderful to have students from your part of the world. We've had students from Kenya in the past, as well as Tanzania and Uganda, I believe, last year as well. So I've been in Tanzania in the last couple of years. I hope to be back there before too long. So best wishes to you, and thank you for being in medical neuroscience.
Thank you, and thank you for the good work.
Okay, you're welcome. Okay, let's go now to Narayan.
Yes, can you hear me?
Very well, thank you. Thanks for joining us.
Thank you. First of all, I would like to thank you for this wonderful course.
You're welcome.
I'm from Finland, so I'm actually a postdoctoral researcher
Yes.
in computational neuroscience.
Mm-hmm.
And I was wondering if you could throw some light on the relationship between the anatomical or structural connectivity and the functional connectivity that we measure with, for example, fMRI.
Mm-hmm. Yes, so this is a very important question, as we're now using these kinds of imaging methods to do lots of research in cognitive neuroscience, as I'm sure you well know, and we need better computational and theoretical scientists such as yourself to help us understand these very complex data sets. So much of the methodology that we have now for studies of human brain function, it's quite indirect, and that's true on multiple levels. So for example, as you very well know, when we do fMRI studies, we're looking at blood flow changes and oxygenation level changes that are only indirectly reflective of neuronal activity, but yet it's pretty good for most assumptions that we would make. It's very, very slow, so that's a limitation of the method, but at least the spatial information is getting better and better all the time. So that's very good. Now, you talked about connectivity studies, and so there are methods now that are basically statistical methods, as I suspect you know. We're looking at the correlation of activity across different voxels within a complex fMRI data set. Now, for me, I'm an anatomist, so I don't particularly like this term functional connectivity, which has emerged as a tool in this field, because it suggests that there are actual anatomical connections underlying the correlation of signal in different voxels of the data. That may be true, but it may not be true at all. There may be very indirect means by which activity in different parts of the brain is modulated in time together. And it may lead to some erroneous conclusions about structure-function relationships. So I'm a little worried about that aspect of the way of studying the active brain using what we're calling functional connectivity measures, when it's statistical correlation. And so I think that's very, very important method, very important information derived from it, but we need to be mindful of the limitations and the possible ways that we interpret these kinds of data. Now, these studies are getting better and better all the time, and so I'm actually quite optimistic that in the future we'll learn even more than we can today with the increased spatial and temporal resolution of these methods.
Yeah, actually, a slight extension to the question and a little more detail about what I am actually doing. So
Yes.
I'm actually using intracranial EEG and magnetoencephalography.
Yes.
And I'm trying to develop methods based on Bayesian analysis to estimate the connectivity from electrophysiological data, which are more direct and have better temporal resolution compared to fMRI data.
Right.
So what I'm confused about is that the conclusions that I draw from functional or effective connectivity, which not only just looks at statistical correlation but also the causal influences between different brain regions, how do I relate that to the underlying anatomical connectivity? And I'm mainly focusing on epilepsy. And I would like to know, in case of focal epilepsies, for example, are there any changes in the underlying anatomical connectivity which leads to this kind of hypersynchronous, which manifests itself as a hypersynchronous electrophysiological activity?
Okay, thanks for getting a little bit deeper with your question. There are certainly going to be changes at the level of synaptic connections and the very structure of the circuits that are sustaining the epileptiform discharges and their propagations as they may secondarily generalize from a focal source of epilepsy. I don't think there will be any wholesale changes in connectivity. That is, there won't be new pathways that will be pioneered and established, and there very likely will not be pathways that will be completely damaged or destroyed. More likely, there will be a change at the synaptic level within existing pathways. So, for example, if you're studying with seizures that have their focus in the hippocampus, the intrinsic circuits of the hippocampus will undergo plasticity, but probably also excitotoxicity. So there likely will be some degradation in those circuits, even as some are strengthened, some will be weakened, some will be lost to excitotoxic damage, and that may happen at a distance as well with the synaptic partners of those medial temporal circuits, structures such as the amygdala, for example, other parts of the ventral medial temporal lobe throughout the entorhinal cortex and the perirhinal cortex, and perhaps even into the piriform cortex at the junction of the temporal and frontal lobe. So we may see evidence of altered structure and function in those circuits, but I don't think you'll see any wholesale changes in patterns of connectivity as a consequence of the seizure activity. I could be wrong about that, but it would be very interesting to see whether you do see such changes, and that might suggest that there may be essentially an additional node, such as a thalamic structure, that may undergo plasticity and then bring into your data set some structure that wasn't previously well correlated from a statistical standpoint.
Okay.
So those are all very interesting possibilities.
All right. And if I may ask one small question, last question.
Okay.
So do you have any insight on these so-called resting state networks? I mean, what are they? And I keep seeing in a lot of papers they talk about resting state network. So what exactly do they mean by this? Because I'm a bit confused. Some papers say that resting state network is like a baseline network and the brain is not receiving any stimulus. but I would assume that we are continuously receiving stimulus. And what is the state of no stimulus?
Yes, so you are correct, and you are right to be somewhat confused because it is a confusing topic. These experiments are typically done by placing someone in an fMRI experiment, and the instruction is do nothing, think of nothing, empty your mind, and simply exist in the magnet as unconcerned and as dissociated as possible from the outside world. So that's usually the instruction given to the subject. And then if you look at what's going on in the brain, there are certain structures whose activity actually seems to increase under those conditions. And many of them are on the medial part of the cerebral hemisphere, structures like the precuneus gyrus, parts of the cingulate cortex, and the superior frontal gyrus. So there are some parts of the brain that if you are truly doing nothing, they seem to be more active than when you're doing something, such as reading text or paying attention to a particular region of space or a particular feeling or sensation in your body. So this idea of a default mode network is simply used to characterize the state of body and brain when you are doing nothing, when you're thinking about nothing, when you're not attending to a particular task. Now, that's easy to say, but of course, if we think about it, are we ever doing nothing? No, we're always doing something. There's always stimuli. There's always a sense of interoception as well as exteroception, as long as we're conscious and awake. So I think that there is something to this. You know, there is a continuum of brain and body state, and that can be modulated by a person's life experience and to some extent their volition. Certainly some religious practices emphasize this kind of internal modulation of brain and body state, and that is likely to manifest as a greater or lesser increase in this default mode network. So thank you very much, Navarayan, and I appreciate your participation in medical neuroscience.
Thank you very much, Doc, and I'm learning a lot from this course. Thank you very much.
Glad to hear it. Thank you.
Thank you.
Okay.
Group C's thing's just been sent out, but no one's showed up yet, so they can stay in until then. So
okay.
They can stay in.
Let's go next to Grace. Grace, are you with us?
Yeah, finally. I've been trying for 40 minutes. So.
Oh, thank you for bearing with us.
Yeah. First of all, like everyone else, I'd like to say it's a tremendous honor and privilege to finally get to talk to you. Your face is so familiar to me and everyone else studying here. And it's so, it's a great honor. I'm surrounded by a lot of very, very skilled and competent professors, but I still say that you come in at the first place. You take the first place. You're the
thank
best
you, Grace.
I've ever had.
Very nice of you to say that. Thank you.
I have a question. My question is with reference to the modification of the neural circuits to early neonatal life.
Mm-hmm.
It's not so much an elaboration as if I don't know if it's possible for you to maybe simplify these concepts about self-organization and the cell-cell interaction and the ocular dominance columns. I have so many questions, but when studying, I think it's most appropriate to talk about what we have upcoming in exams. But from my understanding, it's about how our brain processes the image to the visual cortex. But I still have, I find it hard to get my head around these concepts. So maybe you could simplify it or break it down.
Okay, sure. Well, let me try to say a few things about brain development that might be helpful. So for many decades now, even centuries, we have talked about nature and nurture as important influences on brain development. And given our state of our science, we now can think of nature as being the products of gene expression. So we can think of the nature side of nature and nurture as what is programmed into our genome, and how are those genes then transcribed and their messages translated into proteins that then can affect the structure and function of neural circuits. So that's what we commonly think of as nature. So genetic specification is one way that I like to think about that. Now there's nurture. Nurture has to do with experience. It has to do with interactions of the organism with its environment, or the organism with itself, or the organism with other organisms. So this is a way to modulate or modify those genetic instructions. And most people who work in developmental neuroscience probably think in those terms, that we have nature, which is genetic specification, we have nurture, which is sensory motor experience. But I have learned quite a bit from colleagues of mine who I've been collaborating with in research who come from other disciplines, particularly from the physical sciences, from theoretical physics, who recognize that there are patterns in the natural world that are not specified by genes and are not necessarily shaped by experience. They simply self-organize. And these kinds of patterns have been recognized on multiple levels. So one example from the natural world would be the patterns of sand in a sand dune, or perhaps the ripples in the bed of a river or the floor of the ocean. So there's a kind of pattern there, and that pattern emerges in a dynamical way because of the physical forces that are acting upon the unitary particles. In this case, it would be sand and water, perhaps sand and air, in the case of the sand dune. So these colleagues of mine began to wonder whether the brain might also self-organize in some interesting way, not because there's a genetic blueprint for a pattern, and not necessarily because experience shaped the pattern in a particular way. The pattern simply self-organized. So that's what I was trying to get at by making that point about how self-organization is a very important driver of activity in the brain and pattern in the brain, the actual structure of circuits. So not everything needs to be explained by genetic specification, and not everything needs to be instructed by experience. But because the brain is a dynamical system capable of establishing its own activity and shaping that activity through mechanisms of plasticity, self-organization is possible. Now, you may be wondering, and this is often a confusing point, surely the genes are involved with everything about the structure and function of the brain. And that certainly is true at some level. The point I would make is that what the genome is providing us is the capacity to build active neurons that have plasticity and have the capacity to grow and make connections with one another. But the genome is simply not large enough to account for all of the structure and pattern that we see within the nervous system. And so much of the patterns that we actually observe, such as the structure of circuitry in the cerebral cortex, seems to self-organize. And so it's important to recognize that contribution to brain structure and function if we want to see the whole picture of what it takes to build a functioning brain. So I think I'll stop at that point and see if you have any further comment or question.
Yeah, quite stunning, considering how big the genome is. That it's not big enough.
It's not. No, it really isn't big enough.
No, that's amazing.
We have maybe 30,000 genes, and it would take many millions of genes to account for all of the detail that we see if it were all to be genetically programmed.
Just as amazing as everything else about the brain, then. I really like the analogy to the sand dunes. That breaks it down for me. And yeah, I was even reading your article from 2010.
Oh,
yeah, and I read a lot of those scientific articles, but that one was one of the tougher ones, and I had difficulties understanding this high density. But it's just like it's that part of the pattern that you're describing, right?
That's exactly right. So what I was describing in that paper, or what we've been studying, is the density of the singularities that we see in the visual cortex. And these singularities reflect the organization of the circuitry. So the circuitry is organized to create these pinwheel-type patterns. And we were interested to know how we could describe the distribution of these pinwheels as a way to simplify a quantitative characterization of the cortical networks. So we decided to focus on several statistical parameters. The one that was most important to us was the actual density of these singularities. And as it turned out in our modeling work, the density of those singularities was exactly the mathematical constant pi. And what we found in nature, in the actual brains that we examined, was an average density of 3.14. So there seemed to be a remarkable correspondence between the model and what we actually saw in the real world.
Absolutely stunning.
Well, it
But
was
thank
to
you
us at
very
the time.
much thank you very much for your answer, and I hope my way will cross yours one day. My aim is to end up in neuroscience, and yeah, I'm on my way already. So.
Where are you, Grace?
I'm in Denmark.
Denmark. Okay,
yeah.
Wonderful. Thank you for staying up so late to be with us today.
My pleasure.
Okay, very good. Thank you.
We have a question from the Q&A from Ava. I was reading about a recent research at MIT that seems to discover a neural cluster just outside the primary auditory cortex that responds exclusively to music, and another one that appears to be specific to speech. What is your opinion?
Yes, I saw that paper as well. So there's a question from Ava about some new data suggesting that in the human brain there may be a module associated with the auditory system that responds selectively to music. It seems to me like this potentially is analogous to those modules that are much better known in the visual system that respond to face. So we think about face as being one of the most salient and one of the most important stimuli that have evolved for mammals and primates in particular. And it may be that the same is true for the temporal and spectral modulations of sound that we call music. So they may not be unique to the human species, and if so, then they may be an evolved property that is reflected in the structure of our auditory system in some respect. And those systems beyond the auditory system that build associations in the brain. So I think it's a very plausible idea. I think obviously we want much more direct evidence of music-specific processing modules in the brain. The data that were shown are somewhat indirect, so I think they need to be verified with other methods. And I think it would be quite interesting to try to explore this comparable brain region in a non-human primate where it's possible to make microelectrode recordings and try to explore the cellular or the neurophysiological basis for this observation. But just as a matter of principle, I think it's entirely plausible that there may be music-specific processing units in the human brain.
Marina?
Yes, can you hear me?
Yes, go right ahead, Marina.
Thank you. So I just unmute. It got muted.
Okay, I can hear you now, Marina. Go ahead.
I don't see the camera being on. I just clicked it. Okay.
I don't see you, but I can hear you, Marina. So why don't you go ahead with your question? Yes, I can hear you.
Well, I wanted to say today is a Teacher's Appreciation Day, so since you're an amazing educator, thank you so much from all of us in the community. I wanted to have the camera on because I wanted to do the handshake that I saw you do in the dopamine, because I think it should be our common community's medical neuroscience handshake. You know, the dopamine, how they're fighting against each other when the two pathways go in parallel. So I think I really love that part. So my question today is, this might be a little bit controversial, but male and female differences in the brain.
Mhm.
And I have heard about some things about the number of white matter crossing the corpus callosum, and I have also heard of the amygdala differences in how the pathway to the amygdala is different in the females and males. And that's actually the part that I'm interested about. Is it true that females go through the frontal lobe first before reaching the amygdala to make a decision, and males have a more direct route, not entering the frontal lobe? And what other differences can you tell us about, just a general overview?
Thank you for your question, Marina. I think the issue of gender differences or sexual dimorphisms in the brain is a fascinating one. The more we are learning about these topics, though, the more I think it's important to emphasize that what we're really talking about is a continuum. The word dimorphism is probably not appropriate. I think what we'll discover is that there is a continuum of brain architectures, and there may be gender typical expressions in that continuum, but I do believe it will be a continuum. So I think the way that you posed your question is probably too strict a view of the data, suggesting that males and females may have access to the amygdala through qualitatively different means. I think there could be quantitative differences, but very unlikely qualitative differences. So I think that both male and female brains have connections that are rich and profound between the amygdala and the frontal lobe, as well as many other cortical areas in the temporal lobe and the parietal cortex as well. There may be differences in the efficiencies or the speed or the level of activation of these connections between a typical male and a typical female brain. But again, I would emphasize a continuum. So I think that the data that you're referring to typically come from human studies that have lots of limitations on them. And I think if we can transcend some of those limitations, we'll see that the actual data themselves are typically much more complex.
Thank you very much.
Okay.
Yeah, this is what I wanted to do.
Oh, yes, okay. Thank you, Maria.
Thank you.
Glad we could finally see you. And your
Thank you.
hands. Okay, thanks for your question.
And I guess Arian is last person just popped in.
Okay,
Yes, sorry.
Ariane,
I
hi.
had a little trouble to connect. Hi, I'm from France, and I'm about to undertake some late medical studies, so I'm very interested in this course. I was wondering about fatigue, especially. Do we know from a neurological perspective what is fatigue? For we often talk about psychic fatigue or nervous fatigue. Does it really exist? Are there some neurotransmitters that are involved? Can we characterize it as well as we can characterize physical fatigue?
Yes, I think the answer is yes, but obviously it's very complicated. As your question suggests, it's probably useful to differentiate the mechanisms of sleep and wakefulness from those of the kind of fatigue that you're describing. Now, these mechanisms, they are very much related, I believe, because one of the functions of sleep is to give us an opportunity to recover from the fatigue that we accrue during our waking state. So one key molecular mechanism of fatigue that is getting a lot of attention these days is the hydrolysis of adenosine triphosphate, which can lead to the extracellular accumulation of adenosine. Now, adenosine is a neuromodulator that can affect the activity of circuits that express receptors for adenosine. And one such circuit that seems to be very important in changing brain state is in the basal forebrain region, right at the posterior and inferior part of the frontal lobe. There are circuits in the basal forebrain that are sensitive to adenosine, and when adenosine accumulates to a certain level, which we might consider to be consistent with fatigue, then that circuitry is influenced to inhibit the hypothalamic systems that are keeping our brain in an awake and active state. So adenosine seems to be something of a system in the brain that will lead to sleepiness and eventually the onset of sleep itself. And while adenosine is not limited to this basal forebrain region, it seems to be in that basal forebrain where there's something of a thermostatic-like mechanism that is sensitive to the accumulation of adenosine. So adenosine, I think, is an important hypothesis that I would cite in answer to your question, because this is the principal byproduct of energy metabolism throughout all of the brain. So it's probably accumulating in many different places in the brain, but the most sensitive mechanism that will modulate our brain state is there in the basal forebrain region. So once adenosine reaches a certain level, then we seem to want to very much go to sleep. And, you know, a lot of us are addicted to caffeine in one way or another. And what the theophylline and the xanthines in products like tea and caffeine are doing is that they're blocking these adenosine receptors in the basal forebrain. So while our adenosine is accumulating, some of us are self-medicating in a way that tries to make this circuitry less sensitive to the fatigue that is accruing at the biochemical level through the hydrolysis of adenosine triphosphate. So that's one perspective on your question. There are many others, I'm sure, but yeah. So I think relating the work of the brain to the biochemistry and ultimately the neurochemistry is going to be a key component to the answer to your question.
Okay, thank you. And
go ahead.
Do you think there is some function in fatigue, or is it a? I read something about fatigue being an emotion or something like something useful to protect us. Do you think it's?
I think that's very possible. As we were talking about in response to an earlier question, a lot of very good things happen when we sleep. One of them is the consolidation of memory. And among the many important functions of sleep, we have this capacity to replay patterns of activity that we may have experienced during the day. And as those patterns are replayed, the circuitry might actually undergo plasticity and some active consolidation. So we need sleep in one way to provide us with that opportunity. So fatigue is potentially a very adaptive human experience, and I'm sure it's common to all multicellular organisms, but it's an adaptive experience that allows us to enter this state of body and brain that facilitates recovery, facilitates plasticity, facilitates the consolidation of learning. And as we were talking about dreams a little while ago, perhaps other interesting phenomena that cannot be experienced in the waking state. So, where are you, Ariane, in France? What city?
In France, near Paris.
Near Paris. Okay. I'll be in Paris in late May, early June.
Yes.
I teach a course there near the Bastille.
Okay. Where? In Saint-Antoine, maybe?
It is in a place called the Accent Center.
Okay.
It's a Duke University program that we run in Paris. It's called Neurohumanities in Paris. It's a course that synthesizes cognitive neuroscience and the study of literature and fiction and visual art. So perhaps we'll have a chance to meet for a coffee or something.
Okay, that will be a pleasure for me, and I'll try to join.
Okay,
thank you very much.
Nice to meet you.
Thanks.
Okay, we're just about out of time, but I see Patricia has joined us. So Patricia, perhaps I'll turn to you for the last question or comment. Patricia, I can't hear you. Are you muted, perhaps? I'm sorry, I can't hear you. Are you there now? Oh, I'm so sorry.
Have her type in her question.
Could you type your question into the chat field? And we'll try to get to you that way.
And in the meantime, somebody wants to know why the questions and the quizzes aren't the same between each attempt.
Okay, someone asked a question online while Patricia types out her message. Someone asked, why are the questions not the same between attempts? That's because the questions in the quizzes are drawn from a large pool of questions, and each question is constructed in a way to draw from multiple best answers and multiple distractors. And this was by design because I think one can learn quite a bit from answering questions. And you've probably noticed I'm not giving you a question key because I do want you to wrestle with the questions as an opportunity to learn more. I'm happy for you to take some of your own content questions to the discussion forum and work through them with some help from other students in the class. But when it comes to taking the quizzes, I want to encourage you to actually take them multiple times. And so you can take them up to 3 times without any penalty in your score. But you can take them as many as you'd like. You can take them, you know, a dozen or more times, and each time you do, you will see some difference from the question set from which they're drawn. I actually had someone calculate the permutations for me, and the calculation was that the odds of seeing the same quiz twice are 1 in 14 million. So I think that gives you plenty of opportunity to learn through questions without being overly redundant.
Okay, so Patricia's question was, I wanted to know your thoughts on caffeine being an endosine antagonist in Parkinson's treatment.
So Nick is sharing Patricia's question. Patricia's question is my thoughts on caffeine as a treatment for what condition was that, Nick?
Parkinson's.
For Parkinson's disease. Oh boy, so I don't know much about that particular application, except that in Parkinson's disease, having additional stimulation seems to be of a benefit, particularly when patients are in a down state or an off state relative to their medication regimes. So the problem in Parkinson's disease is a relatively low level of dopamine because of the degeneration of the dopaminergic neurons of the mesencephalon. And the classical conditions associated with Parkinson's disease are really a manifestation of dysfunction in the basal ganglia because of the low levels of dopamine that are synthesized and released there once those midbrain neurons die. So one idea is if we could sustain activity in the cortex through some other mechanism, such as activating nicotinic receptors or blocking adenosine receptors, supporting the activity of cortical networks, there may be opportunity to gather more resource, more cortical activity that might allow for the initiation of movement, the initiation of cognitive programs that will support a higher level of functioning in patients with Parkinsonism. So there may be other similar strategies using other neurotransmitter systems that might prove to be effective. And I guess that's about all that I can say about that. Perhaps Patricia could learn a little bit more through her own investigations and share some of that with all of us in the discussion forum. Okay, so I think we'll have to conclude at this point. We're a few minutes over our scheduled time. Thank you so much, everyone who joined us, and those of you who were just following along either on the Google Hangout Live or just tracking along in some other way. I appreciate your participation. And if you care to review any of this, we will be publishing this Hangout on our YouTube channel shortly, so you can review what we've discussed there. So thank you all. We'll probably do this at least one more time before the conclusion of medical neuroscience. Meanwhile, thank you all for your efforts, and I look forward to your continued strong work as we head now towards the final few weeks of the course. So thank you all, and I'll see you next time.